Welcome to the fascinating world of bacterial genetics. Today, we are going to explore one of the most dynamic and surprising mechanisms in biology, transposons. If you ever thought of DNA as a fixed, unchangeable blueprint, prepare to have that idea completely shattered. We will discover how certain pieces of DNA can literally jump around the genome and why this microscopic gymnastics routine is critical to understanding how bacteria survive, evolve, and sometimes become resistant to our most powerful medicines. Let us dive in. To start, imagine you are reading a massive, heavy textbook. Suddenly, a whole paragraph physically lifts itself off the page and glues itself into a completely different chapter. Not only does it move, but sometimes it leaves a copy of itself behind, meaning the book keeps getting longer and more chaotic. In the microscopic world of bacteria, this magical jumping paragraph actually exists, and we call it a transposon. These are tiny pieces of DNA that refuse to sit still, constantly hopping from one part of the bacterial DNA to another. They are the ultimate genetic wanderers. By jumping around, they can accidentally break important instructions or, surprisingly, give the bacteria powerful new abilities to survive. Now that we have an intuitive picture, let us look at the formal definition. In formal genetic terms, transposons, or transposable elements, are specific segments of DNA that possess the unique ability to change their physical location within a genome. They do not just float around randomly. When these elements move, they recognize specific DNA sequences in the host genome. Once they find their target, they cut themselves out of their original location and insert themselves into the new target site. This remarkable ability to relocate is why they are often informally called jumping genes, and it fundamentally alters the genetic landscape of the host bacteria. Building on that definition, we can categorize these jumping genes into two main types in bacteria. The most basic type is called a simple insertion sequence. This sequence contains only the bare minimum genetic code required to make an enzyme called transposase. You can think of this transposase enzyme exactly like molecular scissors. Its only job is to cut the DNA and paste it somewhere else. On the other hand, we have complex transposons. These are larger and carry additional genes alongside the transposase. Most notably, they often carry genes that protect the bacteria from deadly antibiotics, turning a simple jumping mechanism into a powerful tool for bacterial survival. Let us visualize what this actually looks like at the molecular level. Here you see a schematic diagram of a bacterial transposon. Right in the middle, highlighted in blue, is the transposase gene, the code for our molecular scissors. Flanking this central gene on both sides are the inverted repeats, which act as the handles that the transposase enzyme grabs onto when it is time to move. Finally, on the very outside edges, marked in red, you can see the target site duplication. This is a tiny piece of the host's own DNA that gets duplicated during the insertion process. Every part of this structure plays a crucial role in the jumping mechanism. Having seen the structure, let us understand how that red section, the target site duplication, is actually formed. When a transposon inserts into a new DNA molecule, the transposase enzyme does not make a clean, straight cut. Instead, it makes a staggered cut at the target site, leaving overhanging edges. The bacteria's normal cellular machinery sees these gaps and automatically fills them in. Because of the staggered nature of the cut, this repair process results in a short sequence of the host DNA being duplicated on both sides of the newly inserted transposon. This duplicated sequence is the target site duplication, and it is a permanent scar left behind by the jumping event. This mechanism of cutting and duplicating leads us directly to a mathematical reality. The genome physically expands. We can calculate the final length of the mutated DNA molecule using a simple addition formula. L final equals L initial plus L transposon plus L TSD. Conceptually, this just adds up all the physical pieces of DNA. L final is the total length of the new mutated DNA molecule in base pairs. L initial is the original length of the target DNA before the jump. L transposon is the exact length of the jumping element itself. Finally, LTSD represents the length of the target site duplication. If the size of the transposon increases, the final length of the DNA molecule naturally increases as well. Let us apply this formula to a concrete example. We are given a bacterial cell with a circular plasmid that is exactly 4,500 base pairs long. A complex transposon named TN10, which is exactly 9,300 base pairs in length, jumps into this plasmid. The process creates a target site duplication of exactly 9 base pairs. We are asked to find the final length. We choose our addition formula because we must account for all physical pieces of DNA. We substitute 4,500 for the initial length, 9,300 for the transposon, and crucially, 9 for the duplicated target site. Adding these together, we get a final answer of 13,809 base pairs. Physically, this means the plasmid has more than tripled in size, carrying entirely new genetic information. Now that we have seen how transposons physically alter DNA, we must ask, why does this matter to us? The reality is that bacterial transposons are one of the biggest threats to modern medicine. They are the primary drivers of antibiotic resistance. When a complex transposon carrying a resistance gene jumps from a main bacterial chromosome onto a mobile plasmid, it creates a dangerous situation. That bacteria can suddenly share its new superpower with thousands of other bacteria. If we did not have transposons, bacteria would evolve much more slowly, and our antibiotics would remain effective for a much longer time. They are the engines of rapid bacterial evolution. Because these jumping genes are so dangerous, they are heavily studied in medicine. Understanding exactly how these elements move allows scientists to track hospital outbreaks. By sequencing the DNA of bacteria infecting different patients, researchers can see exactly where a transposon has jumped, mapping the spread of resistance in real time. The ultimate goal of this research is to develop entirely new classes of drugs. Instead of just trying to kill the bacteria, future medicines might be designed to block the transposase enzyme entirely, shutting down the jumping mechanism and stopping the spread of antibiotic resistance at its source. To understand how fast this resistance can spread, scientists need to measure it. This brings us to the concept of transposition frequency. Conceptually, this formula calculates the probability of a single cell undergoing a genetic jump. The formula is F equals NT divided by N0. Here, F is the transposition frequency, which gives us a decimal value representing probability. NT is the number of cells that actually show the transposition event, the ones where the jump was successful. N0 is the total number of cells in the starting population. If the number of successful jumps, NT, increases, the frequency F will naturally increase, indicating a highly active transposon. Let us see how this is calculated in a laboratory setting. A scientist starts with an initial population of exactly 5 million bacterial cells. After an environmental stressor, exactly 250 cells have experienced a transposition event, moving an ampicillin resistance gene. We are asked to find the transposition frequency. We use our frequency formula to set up a ratio comparing the rare, successful jumps to the massive overall population. We plug in 250 for NT and 5 million for N0. Dividing these gives a final answer of 0.00005, or 5 times 10 to the negative fifth power. Physically, this means that for every 100,000 bacteria, exactly 5 experienced this jump. While rare for a single cell, in large populations, this tiny fraction is enough to ensure the colony survives an antibiotic attack. We have covered a lot of ground today, so let us summarize the most important ideas. First, transposons are remarkable, mobile genetic elements that can independently move around the bacterial genome, completely defying the idea of static DNA. Second, they achieve this using the transposase enzyme to cut and paste themselves, leaving behind physical evidence in the form of target site duplications. Third, and most importantly for humanity, complex transposons carry survival genes and are the primary engines spreading antibiotic resistance. Finally, as we saw with our frequency calculations, even though these jumps are rare for an individual cell, they happen frequently enough in massive bacterial populations to drive rapid, life-saving evolution. You now understand the fundamentals of bacterial transposons.